Hello there, my name is Nicky Bradley and this is a short video on G protein signalling and how that regulates heart rate. So what we're going to talk about during this video is the relevance of G protein coupled receptors and how they act in signalling. We're going to explain the process whereby a G protein, once activated, stimulates something called cyclic AMP within the cell, and compare this process with how a different G protein inhibits those cyclic AMP levels. And then we'll go on to look at how cyclic AMP regulates heart rate. So the key thing is that protein hormones and something called catecholamines, which is basically noradrenaline and adrenaline, cannot enter cells. They are hydrophilic. They like water, which means that they are also lipophobic, so they don't like lipids. Therefore, they can't get through the plasma membrane because, of course, the centre of the plasma membrane is made up of lipid. And there are no facilitated transport mechanisms, no transporters to help them through the plasma membrane. Because of this, they need a mechanism where the ligand, in other words the hormone or the catecholamine, binds extracellularly to the surface of the cell, to a receptor, and then activates some sort of system inside the cell. And these are known as second messenger signaling mechanisms. The most common of these second messengers is something called a G protein. The G protein is a peripheral intracellular protein, and it has three subunits. You can see there alpha, beta and gamma. So it is inside the cell, it is intracellular, but it's associated with a specific receptor that is on the surface of the cell, an extracellular receptor. This receptor is known as the G protein coupled receptor, and you may see these initials in various scientific articles, GPCR. The G protein uses energy from something called guanosine triphosphate, GTP. You may be aware of ATP, adenosine triphosphate, because that's usually the energy source that mechanisms use. However, the G protein uses GTP. And it doesn't use this really until the end of its process. So it uses it to reassemble the G protein, to come back together into those three subunits. So how do we activate a G protein? Well, first of all, a hormone or a catecholamine comes along and binds to the receptor on the outside of the cell. And there you can see it sticking up above the plasma membrane with all its little phospholipids. The G protein inside the cell then binds to the intracellular end of that receptor. The alpha subunit, when inactive, has guanosine diphosphate. In other words, it's missing a phosphate and it's missing some energy. So it's going to need energy later. So it gets rid of GDP and takes on board GTP, guanosine triphosphate, which is going to be a good source of energy later. the whole G protein then moves away from the receptor. This is so that other G proteins can bind. Then the alpha subunit moves away from the beta and gamma subunits. And you notice it's taking the GTP with it. While the ligand is still bound to the receptor, and because our first G protein has moved away, more G proteins can come up, associate with the receptor, and become activated. So this can carry on repeatedly and give us lots and lots inside that single cell of activated alpha subunits. So we have our little alpha subunit being activated. Each alpha subunit can be stimulatory or inhibitory. So it can either activate or inhibit. And in this case, we're talking about one of the most common mechanisms involving something called adenylate cyclase. Adenylate cyclase is an enzyme that's bound within the plasma membrane of each cell. And we have lots of them within each single cell. So if we want to activate adenylate cyclase, we need an alpha subunit that's going to be stimulatory. So here's our stimulatory G protein. You see we've called it a GS, S for stimulatory. Comes along, it binds to adenylate cyclase. Adenylate cyclase then takes ATP, adenosine triphosphate, floating around in the cytosol, and it converts it to something called cyclic AMP, adenosine monophosphate. Of course, this releases two phosphates as well. Cyclic AMP is often written as CAMP. However, if we've got a different receptor and that's associated with an inhibitory G protein, when we activate the alpha subunit from that receptor, it has an inhibitory effect on adenylate cyclase. So it's still activated in the same way as RGS, but when it binds to adenylate cyclase, it blocks it. If it blocks it, it's going to stop it producing cyclic AMP. That means that the ratio of activated GS to GIs regulates the levels of cyclic AMP within the cell. Bear in mind we said there's lots of adenylate cyclases, so 50% of them are activated by a GS and 50% are inhibited by a GI. We're going to get about 50% of the possible cyclic AMP. Depending, of course, if the GS then increase, we'll get more cyclic AMP. If the GI increase, we're going to get less cyclic AMP within that single cell. So, heart rate is determined by the steepness of something called the pre-potential. This is just prior to full depolarization within the autorhythmic cardiomyocytes of the heart, specifically the sinoatrial node. The sinoatrial node because this is the first point where that intrinsic autorhythmic pathway starts. The steeper the pre-potential, the faster the heart beats, because each of those depolarizations is one heartbeat. So the quicker we get to depolarization, the more heartbeats we will have within a specific time period. This is governed generally by the autonomic nervous system. The parasympathetic nervous system generally dominates. This is known as vagal tone, because the intrinsic rate at the SA node is 100 beats per minute. If we had that rate all the time, we'd probably have heart failure very quickly. So the parasympathetic nervous system releases the neurotransmitter acetylcholine to slow the heart rate down. Its partner, more fight and flight than rest and digest, the sympathetic nervous system, releases noradrenaline to increase heart rate. So if we look at this pre-potential, the pre-potential is that bit below the dotted line. The dotted line represents something known as threshold. Threshold is just prior to you getting full depolarization. So looking at the pre-potential, we have an influx of sodium and calcium. Influx means that sodium and calcium flow into the cell. And this is through their own specific channels. And in terms of the pre-potential, these are called funny channels. And actually the pre-potential is called funny current. So then that brings us up to threshold. At threshold, those channels are closing, but new channels are opening. So these are also calcium channels, but they stay open longer. They're long-lasting calcium channels. Calcium moves in to the cell, it influxes, until we get up to the peak of depolarization when they close. To repolarize, in other words, bring us back to that point where we started below threshold, calcium has closed, but potassium channels open. Now potassium moves out of the cell. If you want to know why, you need to go back to resting membrane potential. But the calcium has closed now, we've stopped that influx, but now we have potassium efflux moving out of the cell. This brings us back down below threshold, back to our starting point, which is about minus 60 millivolts. So you can see there that's quite a long slope. And if we carry that on with more heartbeats, in other words, more depolarizations, we can see that the heart here is beating relatively slowly. Our peaks of depolarization are quite far apart. Conversely, if we look at what happens when we make that pre-potential steeper, we get these beats coming together a lot closer. And here, over this specific period of time represented here, we had 4 heartbeats in that space, as opposed to 8 in the one below. Because if you look at that pre-potential, it's much steeper. So this is where we're going with our levels of cyclic AMP. We are going to change the steepness of that pre-potential. So going back to our neurotransmitters, in the regulation of heart rate, cyclic AMP levels within the cell are dependent on the neurotransmitter availability outside of the cell. So here we have a beta-1 adrenoreceptor, and you've possibly heard of beta blockers. This is what they block. A beta-1 adrenoreceptor binds noradrenaline. Happens to also bind adrenaline, but that's another issue. Noradrenaline is released from sympathetic neurons. Noradrenaline binds to its beta-1 adrenoreceptor, so this is like our ligand before, and it activates a stimulatory G protein. So we go through that whole process, G protein moves away, alpha subunit moves away, and the alpha subunit goes off and activates adenylate cyclase. Adenylate cyclase takes ATP and converts it to cyclic AMP. So this is going to drive the levels of cyclic AMP up. Cyclic AMP then sensitizes our sodium channel and calcium channel, our funny channels that are involved in the prepotential. If they sensitize them, it means that these channels open quicker. If they open quicker, we're going to get more of these ions, sodium and calcium, into the cell, and that's going to make that prepotential shorter and steeper. So it's going to drive heart rate up. Conversely, if we have, as we usually do in the same cell, cholinergic receptors, M2 cholinergic receptors, or muscarinic receptors as they may be known as, they bind acetylcholine. If you remember, acetylcholine is released by parasympathetic neurons to reduce heart rate. So acetylcholine binds to its M2 receptor, but in this case it activates an inhibitory G protein. So we go through the same thing, alpha subunit's activated and off the alpha subunit goes, but when it binds to adenylate cyclase, it blocks it. And if you remember, this may not be the same adenylate cyclase, we have lots of them within the cell. So it will block adenylate cyclase when it binds to it, which means that adenylate cyclase can't make cyclic AMP, so it will drive those levels down. This will reduce the sensitization and reduce the opening of our funny channels, of our sodium channel and our calcium channel involved in the prepotential. And you can see there that they are effectively closed. If they don't open for a little while, the prepotential will be longer. The slope will be longer, shallower. And this, of course, will drive those depolarizations and therefore the heartbeats apart, slowing the heart rate. So if 50% of our adenylate cyclases are blocked, we're going to be 50% down on our maximum possible cyclic AMP. However, if we activate the beta-1 adrenoreceptor with noradrenaline, that's going to drive cyclic AMP levels up. And it is just like that. It's not an on and off thing. It's like a seesaw. It's like a constant changing in these levels that is going to change the steepness of that prepotential slope and therefore change heart rate. So some other things G proteins can do, just to show they can do other things. They can activate something called protein kinase A. Protein kinases generally phosphorylate things. In other words, they take phosphates from the cytosol and they add them to other things. If they phosphorylate channels, generally this leads to the channels being sensitized, opening more quickly or staying open longer. If they phosphorylate enzymes, this can be excitatory or inhibitory. So we get a complete snowballing of effects due to phosphorylation. G proteins can also activate another enzyme called phospholipase C. This is membrane bound. It uses phospholipids to make two things. So it makes diglycerol, which then goes on to activate something else that phosphorylates. But it also produces IP3. IP3 binds to receptors on the sarcoplasmic reticulum or endoplasmic reticulum, where there's a lovely store of calcium. This releases calcium into the cell, and this can be used for contraction, for, if it's a contractile cell, for exocytosis, where the cytoskeleton moves vesicles, or if calcium combines with something called calmodulin, it can lead to signaling. So again, an explosion of things happening. Through adenylate cyclase again and cyclic AMP, G proteins regulate the release of ions into the small intestine, the ileum. Because ions are released, water will follow. So the number of ions released regulates the amount of fluid moving into the small intestine. So if you overstimulate adenylate cyclase, you get more ions, you get more fluid, and you get diarrhea. And funnily enough, coffee can do this and various other things, bacterial toxins, all sorts of things. So it's quite interesting in terms of regulating that fluid and the pathophysiology behind diarrhea. So let's deactivate it. Okay, so ligand is no longer bound to the receptor. Cyclic AMP is deactivated by another enzyme with a wonderful name of phosphodiesterase. The alpha subunit, this is where it uses that GTP, remember, that it's been carrying around all this time. It hydrolyzes that, in other words, it breaks the last phosphate bound, and this releases energy. This energy it uses to return to where it left beta and gamma, so that the whole G protein can come back together and hang around until when the next ligand binds to its G protein coupled receptor. Well, thank you for watching. I hope you found it useful and informative, and I'll perhaps see you on one of my other videos. Bye.